And thank you all for attending this talk. Today I'm presenting a project that was part of my PhD dissertation in the University of Oxford, and as was said, it was lucky enough to be published in LabOnAChip in last year. During the presentation, I will briefly mention and introduce what fluid walls are, which is like the big topic of this talk, and presenting their properties and their benefits over or comparing them with standard microfluidic technologies. And then finally, for the sake of time, just presenting the qualitative side of the results that we obtain, attempting to recreate the cortex striatum circuits of the brain inside of a dish, and with functional assays on axonal regrowth that really exploit the properties of fluid walls at the best. So we all know that microfluidics is nothing more than a miniaturized hydraulic system, where the aqueous phase is confined inside solid pipes, usually made of glass and or some sort of plastics. The most common is PDMS. However, at the microscale, there is not a real need to confine liquids inside solid pipes because interfacial forces are dominant over gravitational ones. So following this logic, we can take one of the most common vessels used in biology, like a petri dish. We can deposit nanoliter or microliter droplets inside the dish and overlay with an oil just to prevent evaporation. Well, these tiny droplets can be considered microfluidic environments per se, where the liquid-liquid interface between the water and the oil, that's what we call fluid walls. This has some benefits, including using only materials that have been widely used by biologists in the last decades, without need of any other additional materials that was not part of biology before. We never have air bubble failures, simply because air bubbles can float away and burst into the atmosphere. And we can have direct access to our cell culture everywhere inside the microfluidic device without need of annoying or complex inlet and outlet designs. So for instance, we can pipette different reagents either by hand or using like a fluid handler everywhere over our cultures. And finally, even probably more relevant for today's talk, we can reshape the microfluidic circuits above live cells without affecting their availability at all. I talked about depositing droplets, but actually we can do a lot more than that. We can take a petri dish, as I mentioned before, cover the bottom of the dish with a very thin layer of cell culture medium, and overlay with an immiscible fluorocarbon called FC40, which is totally bioinert. It's permeable to gases and it's already used in microfluidics. And then with the use of a nozzle attached to a 3D traverse, we can jet additional FC40 that sweeps the underneath medium layer away from the dish and pinning the FC40 to the bottom of it. In this way, it's dividing the aqueous phase into small pockets or into small microenvironments. Here's a video that shows exactly what I just described to you. We can see the traverse that is moving a nozzle up and down. The medium layer is stained with blue dye just to enhance the visibility. And this helps us visualize that where the jet has passed is leaving a white trace behind it. And that is the FC40 that is pinned to the bottom of the dish. Speeding up the process, it seems that here we're making like a checkerboard made of 256 independent environments. It's quite understandable that just changing the path of the nozzle, we can make any two-dimensional circuit in minutes inside standard dishes that are already used and available in every biological lab. So coming to today's talk, the challenge of this project was to exploit these fluid walls properties to start creating a brain inside of a dish. And the more we talked with neurobiologists, the more they agreeing, saying that the main characteristic of the brain is to be made of different functional areas that are unidirectionally connected in electrical circuits. This is simplifying it a lot, is due by the shape of and the characteristic of the neurons that collect the electrical information in the dendrites, elaborate in the soma, and then send it along the axon, and never vice versa. So in order to capture this directionality, we started designing a very simple microfluidic design which is made of shaped like a dumbbell, made of two chambers, one containing cells and the other one containing, for the moment, nothing, just connected by a little channel. And here you can see the experimental setup that we use, where we have 21 independent dumbbell circuits inside a single 6-centimeter dish. Once again, stressing from the side view, we can see that the only solid boundaries ever present inside this system is the dish itself. Because we don't have any solid boundaries, we need to make sure that when we deposit cells somewhere, so they actually remain where they have been plated and they do not move or flush away. We've done that with a fluorescent lentivirus transfection, where we proved qualitatively after 2 days that no cells have been moved into the distal chamber nor inside the channel, but actually they attached exactly where they've been plated. And after about a month time, we can see that all the neurons have grown, the ramifications invading the channel, and then the distal chamber, creating these nice arborized branches, but yet again, no cells, no somas, to be more precise, can be detected in the far chamber, just a couple of 100 microns inside of the channel. The second step was to prove, because we want the electrical signal to be able to travel just from left to right, is to prove that only axons can actually grow that far to go into the distal chamber. And we did this using immunostaining, proving that dendrites in red, they only grow in the surroundings of the somas that are highlighted in blue, while the axons in purple are the only one that can grow far enough to go and invade the distal chamber and go through the channel. At this point, we started thinking we can connect the two sides and having two different populations. So if we only have cortical cells on one side, we can now put striatal neurons on the other side. Again, with immunostaining showing that in the left-hand side chamber, we only have green positive cells, classic marker for cortical neurons, while in the right-hand side, we have a DARP 32, or yellow in this case, markers that indicates the striatal neurons, which are, as you can see in the merge panel on the bottom right side, they are entangled with the green axonal terminals, creating synaptic connections, which are unidirectional, as I stressed a lot before. And so, again, this is very preliminary results, but we have the impression that we have inside normal vessels that are widely used in any biological lab, we have this unidirectional connection between the striatum and the cortex, exactly as it happens in vivo. In the last few minutes I have left, I'll present to you this functional assays that we developed. So we can take our cell culture at about a month time, as I said before, with or without any cell type in the right-hand side. And we can, simply removing the FC40 overlay and flooding the dish with additional medium, we destroy the fluid wall. So we destroy the microfluidic patterns, and we just leave the cells attached to the bottom of the dish and alive. We can then, using a submerged jet of medium, we can create a targeted damage around any point, let's say in the channel or in our culture, and creating, in this case, an assay that forms a contactless and chemical-less axotomy assay. And probably even more nice than anything else is that we can once again recreate around the damaged cells a new cell circuit, a new microfluidic circuit. And we only were careful of creating the fluid walls just slightly offset compared to the original boundaries in order not to damage the cells that were living on those boundaries. So we prove again that the cells, after a month's time, they were like very stable and they remained alive before and during the damage. We can destroy the microfluidic device and recreate it on top of it without affecting cell viability whatsoever, apart from the area where the damage has been done. And because we can recreate our microfluidics around live cells, we managed to observe the regrowth and hence the repair of the wounded area around that. With that, I think I ran out of time, so I would like to thank you for your attention. As I said many times, for the sake of time, I just presented qualitatively what this project was, but if the publication contains a lot more data on, more quantitative data on how we manage the flow between the two chambers of the dumbbells, and the outgrowth of the axon and the regrowth against different molecules and against in different conditions. Once again, thank you, and thank you for the invitation, and thanks to all the people that collaborated on this project.
So thank you very much, Angela, for introducing and inviting me here to that session. I think it's very nice to connect the whole world with one session. That's cool. So as Angela said, we work on, let's say, synthetic cells, and it's the term matter to life, which we also have a Max Planck School on, matter to life. So it's a master PhD program, which is a national program in Germany where a couple of now close to 80 or 90 colleagues are collaborating on that topic in a very broad sense. Give you a kind of interest. So what we are in the group is interesting in, and let's say we worked a lot in understanding, analyzing cellular systems from a biophysical point of view and biochemical point of view in the past. And of course, there's lots of known about cellular systems. And with this knowledge, we now try to design like life systems. So the question, I'm a physicist by training, comes, can we build something like what we see under the microscope in the real world? And of course, the complexity is huge, and we may never succeed in constructing a full cell, but maybe just parts of it, certain functionalities. And that's what I want to bring up today. To give you an idea how a Max Planck department operates, it's a department which has several groups, independent groups. So it's my group. Then we have Heike Böhm. She's working on biophysics of cellular interactions. Ada was a group member. She is now a professor at Bayreuth University here in Germany. She works on growth factor mechanobiology. Smriti worked on organoids. She's now with a company, and Ilya, he's still here. He's our experts in microfluidics, and I will also talk about work which we do together with him. But there is more to give beyond the department itself. The Max Planck Society also offers the best postdocs which once worked in the group to become a head of a Max Planck partner group. So these, you see here, 5 former postdocs of my lab, which are now back in their home countries, having a position there, and they can get a grant and this title, head of Max Planck partner group, for I think 5 + 5 years, at least for 5 years, and continue to collaborate with us or with other colleagues at the Max Planck Society. So for them, the financial aspect is little, but I think the connection which we can keep is excellent. But now let's come to the science. So we try to build synthetic cells since more than 10 years, and we started it originally by a very traditional biochemical approach. So taking lipids which form then a giant unilamellar vesicle, and you include then via fusion with small vesicles, transmembrane receptors. And at this time, we were very much interested in the receptor integrin. We're looking at clustering of integrin receptors if a cell is approaching a tissue. And we wanted to build something like this. So a vesicle, very primitive, a vesicle which eventually can bind to tissue. And this was partly successful, but the main problem was it was not really reproducible. So you get images like you see them here. You see a side view image on a surface, cross-section of a surface, and you see these vesicles adhering against the tissue. You see it's a flat interface. In blue and in green, you see the lipids, and in blue, the integrins, which are labeled. And what you see here could be partly reproduced, but it was difficult to actually now increase complexity by adding other molecules. And it's mainly the point that these GOVs are mechanically too weak. So whenever, more or less, when they see an interface, it's falling apart. And if you now think about manipulating them further, it's very difficult. It's not totally impossible, but very difficult to get quantitative reproducible experiments. So this led us in a kind of dead-end road where we nearly were at the point saying, well, let's stop this. We are not there. We cannot do that, what nature can do. But then in a side project, and also that is when Ilya started, we got hands on microfluidics, and I think we had an ideal introduction to microfluidics. So this is droplet microfluidics, where you see it in many, many labs all over the world, where you have 3 channels meeting each other. One is filled with water, the other 2 with oil. And then you see the oil phases are cutting out droplets. And these droplets are stabilized in this oil phase because they're stabilized by a surfactant. So the surfactant in this case, for instance, is a tri-block copolymer, where the oil phase is interfacing with the perfluorinated polyether, while the water interface is interfacing with polyethylene glycol. And this is now reducing the surface tension between water and oil, and that's why you see that these droplets are touching each other, but they do not fuse. So they do not and so mechanically, from a mechanical point of view, that's ideal. It's now a very stable water droplet compartment, which we were missing with the GOV approach, which I presented before. But then it's the question, so how do you get something into these droplets in order to build up something which is a cell-like, for instance, you would like to have a lipid bilayer, and you would like actin to a cortex coupled to the lipid membrane. You would like maybe have to transmembrane receptors and so on. And eventually you even would like to have it outside the oil phase, so getting it into physiological conditions. But for the first question, how to get something inside, Ilya developed this very nice sequential pico injection microfluidics, where you see here now a droplet passing, and this is very much slowed down, this video. And the side port is injecting this black liquid into this passing water droplet. And this is a little bit on overpressure. It's not so much that you press in droplets into the oil phase, but it's enough pressure to press water into this passing droplet when there is the electric field of these electrodes are turned on, because the electric field are generating micropores to the membrane, and it's like microporation, which you might know from cell biology experiments. So this is nice because now you can control exactly how much liquid you inject into these passing droplets. It's very reproducible. You can do it stepwise. You can think about having several of these side ports which are operated individually, and that really allows you to bottom up inject molecules and build up a cell according to your design principle. And that means you could, for instance, start with a lipid bilayer first, and then you could include small lamellar vesicles with transmembrane receptors or other molecules, and then you see how they assemble. And that was eventually, so this technology, a game changer for us for building up synthetic cells or functions which are originating from natural cells and lead to different publications. And just to show you some basic results, so we then came back to the adhesion problem, getting integrins integrated into these lipid vesicles inside, and eventually you want to have it then out in the aqueous phase in a physiological environment to adhere, to make these spheres adhesive against a tissue. And you can now destabilize these mechanically important, at the beginning, mechanically important walls by adding here small molecular surfactants, so they are smaller in molecular weight, and through entropic reasons, they integrate then into this stabilizing layer. It's destabilizing it, and then the surface tension is pulling out these finished droplet into the aqueous phase, and then you can make, for instance, adhesion against fibrinogen. And that's what you see here. You see now these droplets making a very nice adhesion, reproducible, very stable, and you can even then add cytoskeleton or other protein functions step by step, and see eventually how the cell can work. And there's just a few examples. First of all, this is a triple side port, which each of these side ports can be addressed by these by these pairs of electrodes, and you can inject then one molecule or one chemistry one by one. And this leads then to different applications. I'm not going into details here, just showing you what could be done. This is an example where we included tubulin microtubules and kinesin motors and forced them to form spindle formation. This is where we then use the cytoskeleton to, upon deformation, that you start to look into deformations. Eventually, can we get here some division? We were not really getting this reproducible, but you can look into division problems of droplets. This was a very intensive work here on the left side. We included here, first of all, this cytoskeleton, actin cytoskeleton in a cortex form. We also included here myosin as a contractile motor. And then we needed energy. Energy, that means you want ATP, you need ATP. And this ATP were generated from ADP by including here ATP synthase in this included droplet. It has an ATP synthase. And as you may know, this enzyme, in order to be fueled, it needs a proton gradient. And the proton gradient we generated by bacteriorhodopsin. Bacteriorhodopsin pumps protons across the membrane, and such to generate a gradient between the inside and the outside. And this gradient is then seen by the ATP synthase and then generates out of existing ADP ATP. And the bacteriorhodopsin functions by absorbing light. So you shine light, and this generates an ATP, and the ATP fuels the myosin, and then you see contraction. And that's exactly what you see in this movie. So you shine light, you generate ATP, you see contraction, and that's a nice control. And eventually, if you combine now adhesion with this contraction and you control that, you can make these droplets also to move over the surface, stimulated from outside. So while this is maybe some very basic and strongly engineered applications, we then went to more biomedical inspired applications to use synthetic cells. And one is, we are looking now these days very strongly into synthetic immune cells. So can we generate synthetically cells which are having functions of immune cells? And a very basic concept is that you combine this from cytotoxic lymphocytes, the cytotoxic ligand, with the antibody presented by B cells, which gives you high selectivity. But this is somehow done in CAR T cells therapy, but CAR T cell therapy is, of course, while it's functioning, it's applicable and it's successful, but it's very, very expensive and will never be applied, I think, to all cancer patients because it's too expensive. So how can you make this maybe simpler? And that means combining now this antibody with the cytotoxic ligand on a simple synthetic immune cells. And in principle, we can do that. So we generated these droplets, immerse them into physiological conditions. And you see now the yellow ones are the synthetic immune cells with a antibody against a tumor cell line and the cytotoxic ligand. And if you then follow this, you see that this green droplet attaches to the tumor cell and sends it into apoptosis. You see the death signal is somehow associated with the red color of this tumor cell. So in principle, these synapses can be formed. They also generate deaths to specific cell types. But if you then eventually go in vivo, you see immediately now after this is now these synthetic immune cells injected into, into mice, even after 2 minutes, 2 hours or 10 minutes, more of all of these vesicles are at the end accumulated in liver. And not really in the other organs or in blood. That means it's simply cleared by the immune system and it's identified. It doesn't allow you circulation times in order to find the target. So it's a failure. So how can you overcome this failure? So first of all, this is a summary that we can generate the synthetic immune cells by a very simple method. You see killing, but the problem is the short circulation times and also reduced targeting, also partly due to reduced circulation time. So how can we overcome this? So the long circulation times is that you replace now this GUV with something which circulates strongly in our system, in humans or in animals, which are red blood cells. Yeah, I will come to that. And the second part, which is important, is that the immobilization of the antibody or the death ligand on the surface of these droplets need to be very stable. In vivo, it's a very harsh environment, so we have different kind of ligands which are competing with this immobilization, and you need to stabilize this strongly. And this is a chemistry we developed. It's based on HISTAC NTA chemistry. You know, proteins can genetically engineered with a HISTAC, rather straightforward, and you can use HISTACs with a nickel 2+ ion to make a complex with NTA. You use this for purifying, for instance, and there the complex can be released by counter ion, by counter ligands on purpose to get rid of the columns. But here you need it very stable, and this is possible by replacing nickel 2+ with cobalt 3+, and I will come to this chemistry. So it's very important. So long circulation times and stable linkage of ligands on the surface of these red blood cells via this cobalt 3+ chemistry. So first of all, red blood cells you take from, ideally you can take it from patients of interest. Eventually you take out the hemoglobin to have the reservoir inside empty. Then you have, that's why it's called ghost red blood cells. And then we functionalize them with lipids, which are carrying an NTA. And the NTA is then ready for grabbing proteins which carrying a HISTAC. And then you have an immune ghost red blood cells which have the ligands of interest on the surface. Now, to show you the importance of operating with cobalt 3+ instead of nickel 2+, so we immobilize FAS ligands or trail ligands, which are these proteins which are killing cells, introducing the apoptosis signal. And if we do this in a petri dish, then and include nickel 2+ with FAS ligands or trail, you see a response. So you see here that there's a fraction of the cells are sending in apoptosis, but a larger part is not really sending into apoptosis. And this is because most of these ligands, even in the petri dish, are lost on the surface, from the surface, because of counter interactions. Now, if we switch now from nickel 2+ to cobalt 3+, then you see there's a strong boost. So with trail, you even get 100% killing now. Also with FAS ligands, it's to 50%. So it's really substantially enhancing the cytotoxicity of these now synthetic immune cells. So red blood cells equipped with a death ligand. Now, if we then go into back to mice and we circulate even for 24 hours or 48 hours, you see that there's a large distributions of these ghost red blood cells now in liver, lung, heart, a little bit in spleen and kidney. So you get long circulation times. These ghost red blood cells are still functional and active. So you solve circulation times to find the target, and eventually you can induce killing. These killing experiments in mice we are just currently doing in experiments. To summarize this, so we have here, we can we take the red blood cells, we make them empty, we functionalize via the cobalt-3+ chemistry, ligands of interest. We can even then include microRNA or mRNA as a therapeutic or any therapeutic drug, which then goes inside and then can be applied in vivo applications. Now coming to this cobalt, to this HISTAC NTA or IDA chemistry, which I think is interest for many applications where labeling on surfaces needs to be stable. And you could think also about just simply labeling with fluorophores if you want to keep them under harsh conditions on surfaces. And the problem, well, HISTAC is very nice because any protein can be equipped with genetically with the HISTAC and with nickel 2+, but the problem is that the stability fails with nickel 2+. So we need to replace this. And we have a protocol for this. I will be happy to share this with you if you're interested in. You induce here a cobalt carbonate, a salt, which replaces now the nickel 2+ with a cobalt 3+. So if you're interested, we can send you even the cobalt carbonate. It's a salt which you cannot easily buy. And we can send you the protocol, and it's really easy to apply. And the result is really superior to what you have with nickel 2+. So here you see if you do labeling with nickel 2+ proteins via nickel 2+ and then add one of these counter ligands like IDA or EDTA or DTT, they simply delete this complex and you don't find any signal. That's the blue one. But if you do it with cobalt 3+, you maintain the stability completely, and you can even keep that for many weeks stable or days. So it's a very stable complex, and it has the stability of a covalent bond. It's not a covalent bond, of course, but it has the stability. So it cannot be, it makes really difference in functionality. Now, if I still have time, I don't know. I didn't see the hand from Angela yet. So we then explored this aspect further, that we went to fully synthetic extracellular vesicles. Now, oops. So extracellular vesicles is a phenomena which is known from natural cells, that they release vesicles which contain mRNA, they contain surface proteins. It's not really clear actually known if they are programmed or not, and but it's identified as a very important signaling process of cells if it comes to signaling over distances. So if this cell wants to signal on the cells at a certain distance, the exosomes send out these microvesicles, and then you induce signals on the recipient cells. The problem is that these extracellular vesicles are very complex, they're very diverse, and it's very difficult to get a quantitative understanding. But it would be nice to make such extracellular vesicles really to engineer, to make them quantitative, reproducible, and then induce very specific, precise signals on the recipient cells. So that allows you really to understand also basic signaling processes in a complex environment. This is already, these natural extracellular vesicles are already used in diagnosis and therapies. There are companies which are isolating natural extracellular vesicles and applying it as for diagnosis or for therapy. So it's of great importance. And the question is, can we improve this even by mimicking these natural ones by synthetic ones, so reducing limitations like the fluctuation of the product conditions. It's very also time consuming, very expensive to isolate them. And of course, error-prone isolation is foreseen. Now also scale up is possible. So you can either make it super precise using microfluidics, or we use a in vitro assembly, which is really a kind of, you shake the system and then you get up synthetic extracellular vesicles. But what you always need is, you need to have the information what mRNA are of interest and which surface proteins are of interest for a certain therapeutic, for instance, therapeutic approach. If you have this information, then you can really build these synthetic extracellular vesicles very nicely. You see here the lipid membrane. Here we immobilize CD9. We included microRNAs. And if you compare natural extracellular vesicles with the synthetic ones, you don't really see a difference. We have a very precise size, for instance. Here, this is about 1 micrometer, while the natural source is very diverse, smaller ones and larger ones. And probably the composition is also diverse. So it shows you just the physical creation of these extracellular vesicles is reproducible. They behave similar. And now if you have then, if you go into applications like wound healing, then there is already known that in wounds you find these natural extracellular vesicles and that they play a role in wound healing. And there's information in literature that there's a certain mRNAs in these extracellular vesicles, certain lipid compositions, and certain surface proteins. But it's, of course, it's a number of microRNAs and surface proteins playing a role. But with this information, you can start to build, and you can now vary the composition and engineer it and see how it's impacting wound healing. And just to start here, you have the initial wound. That's a skin model. You introduce a wound. That's the wound, and then you see how in one day how the wound is shrinking. If you do it with human serum, that would be the natural healing. You see the wound is shrinking from this size to this size. Now, if you do it just with buffer, you see that the wound stays larger than in the case with human serum because important signals are missing. Now, if we add to the buffer treatment our synthetic extracellular vesicles, you see also the wound is shrinking in the same way than with human serum. And that's in principle shown here by this graph. But you can go now further on. You can now, for instance, look at proliferation of keratinocytes, one aspect of wound healing. And by creating now different combinations of microRNAs and surface proteins, you can see that the response of the keratinocyte proliferation is very different. And you can now pick the conditions where the proliferation is best. And this is something which you never can do with the natural extracellular vesicles, but with the synthetic approach, you can really engineer and boost the eventual signal. You can even go further. You can now do a genetic analysis of signaling paths, which signals, so you know exactly what you offer the cells, what proteins, and then you can investigate the signaling paths which are introduced. I'm not going into these details. We are also not best in the genetic analysis, which we do always in collaboration. But I just want to give you this idea. So if you have a extracellular vesicles where you know exactly what you offer, you can identify very precisely signaling paths, which otherwise you could never study because it would be much too complex and not clean enough. The other last example is atopic dermatitis. Atopic dermatitis is also an immune disease, which is very painful to patients having it because the skin breaks and it's like really the skin is burning. It's a chronic inflammatory skin disease. It's many people suffering from that. It's a high burden. But there's also a treatment. One treatment is that you simply use cortisone. So what's happening with atopic dermatitis is that the skin breaks. It enhances then cytokine release from the immune cells, from the helper T cells, and the release of cytokines even makes the migration of the keratin stronger and the break of the skin even more severe. So it's a kind of circle, self-enhancing circle, which only can stop if you calm down the immune cells not to release more cytokines, and that's usually done by cortisone, cortisone treatment. Cortisone has disadvantages. Well, it helps, but after some time, your body gets used to it. The response is reduced, and it has some side effects. So you would like maybe to use something which is more natural, and then you find in these wounds also natural extracellular vesicles. And again, you analyze them, what surface proteins these have, which microRNAs, and what is the lipid composition. You can build them synthetically, and then you can apply them. And here you see an example. So you see a healthy skin. So that's the dermis, that's the upper dermis, and it's well connected. And if the disease model by adding cytokines is initiated, then the skin is really falling apart. Yeah, there's no clear interface between the epidermis and the dermis. Now, if you take this situation in the disease model and now treat it with the synthetic extracellular vesicles with a certain composition, but cytokines stop, then the wound is, or the skin is reconstituting again. And even if you keep on adding cytokines, then even then the synthetic extracellular vesicles are overcoming this immune response. And at least in the skin model, it seems like to be a reasonable way to treat atopic dermatitis, to calm down the immune system. And as I said, this tuning of the composition is here very essential, which can be done by synthetic approaches. That's again, we look into, and this is very preliminary, we look now into different genes which are up and down regulated to understand the signaling processes, but there's not really a clear picture yet. In the end, I think I stop here. I think I named a few, or showed a few people who are working on this. We started this many years ago with Benny Geiger from the Weizmann Institute in Israel. We have a center, Max Planck Center with Bristol University together, where we collaborate. And these are people who are involved besides others. Ilya, I mentioned, he very much develops this microfluidic technologies in our group. Amelie Bank, she's developed the cobalt-3+ chemistry now together with Julia. And Oscar is a super PhD student. He now has his own group in Saarbrücken working on extracellular vesicles or also synthetic immune cells. And then I thank you very much for your attention.